Hello everyone, and welcome to today's webinar, Applying a Closed, Modular, Semi-Automated System to CAR T-Cell Therapy Manufacturing. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more, visit thermofisher.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Yongchang Ji, Ph.D., Process Development Manager in the Cell and Gene Therapy Group at Thermo Fisher Scientific. Dr. Ji, you may now begin your presentation.
Hi, my name is Yongchang Ji. I'm the manager of Cell and Gene Therapy at Thermo Fisher Scientific. Today, I walk you through Thermo Fisher's workflow solution for a closed modular semi-automatic system to CAR T-cell therapy manufacturing. Because the novelty of the cell therapy field and the inherent living cell characteristics of the product, the field facing a lot of challenges from management of supply chain, logistics, to bioprocessing, how do we scale up and scale out, and all the way to consistency, how do we manage the quality and performance of the product. And plus there are numerous new technologies rapidly appear and people applying to their processes. Now, if I were to summarize all these challenges, all these are boiled down to the manufacturing. And there's a saying, in autologous cell therapy field, the process is the product. Let's look at some of the manufacturing issues facing autologous T-cell therapy. On the left here is a typical workflow of the T-cell therapy, and it starts with patient at hospital collecting the blood in a process called leukapheresis, and then this blood will ship to the manufacturing facility, which is a different location normally, and T-cell gonna undergo manipulations, modifications, and then this modified T cells will then ship back to the hospital and get administered to the patient. If you look at the whole process, it's rather very academic and a lot of hands-on, labor-intensive, and people use a lot of open processes, and this process is very complex, and it uses a lot of different products, and the instrument applied in this process normally are not flexible to be used in other processes. So this is where we think to solve all this problem, a closed modular and automation system will help to facilitate. And Thermo Fisher Scientific has the largest scientific tools collection in the world. And we ask ourselves, how can we contribute to the cell therapy field? When we look at internally, we have the largest collection of reagents and protocols, and we have existing cell processing platforms and the new technology that accompany with that, and we have world-class analytical in-process tests. Also, we have the capacity of digital sign to integrate the whole thing. And we think, why can't we integrate all these capabilities into one workflow to serve the cell therapy? And by doing that, we create modules of unit operations. Each unit or modules can operate independent of each other. However, on top of each module, there is a software library connected to what we call the brain, which is the DeltaV system. So by using DeltaV, we can connect these individual modules into a whole workflow solution. Now I would like to introduce our Thermo Fisher solutions step by step. Here's the illustration of a typical 1-to-1 process. So the leukapheresis product arriving at the manufacturing facility, they got washed, and then T cell are selected. And these selected T cell are then undergo activation. After certain period of activation, there will be a CAR gene transfer into these T cells. The gene transfer can be done either by lentivirus or electroporation. For today's seminar, I will focus on the electroporation part. And after the gene transfer, these T cells are expanded to achieve certain cell number in order to dose the patient. And then the expanded cells will be washed and formulated, fill finish to ship back to the hospital. And in the whole process, we identify several customer pain points, namely the PBMC isolation step and T cell isolation step. How do we prepare cells before electroporation, and how to achieve good gene editing efficiency. And I will illustrate in this pain point one by one in later of the talks. The first step is normally a PBMC isolation step, because normally the incoming leukapheresis product contains DMSO that need to be washed away, and also other factors, including the platelet or red blood cells that might interfere with the later isolation step, which also need to be washed away. To solve this problem, we invent the Rotea system. It is closed and automated. It has small footprint and comes with its own single-use consumables. On the right here, we see the Rotea cell processing chamber. Rotea, the underlying principle is a counterflow centrifuge, which means we introduce a flow force that against the G-force. Once the flow force and G-force equilibrate, the cells are floating in the chamber and create a fluidized cell bed. And you can imagine with different cell size and buoyancy, we can easily separate different cell populations. And in this case, we are seeing a small cell bed are forming at the tip of cone. And as we load more cells into the cone, the bed will grow much bigger. This is some of the example we use Rotea to do PBMC isolation. On the left here, top is before Rotea processing. We have a lot of red blood cells. After Rotea processing, we have on the bottom here, we have minimal red blood cells, and the T cell proportion got enriched. Not only that, we actually grow those cells to the right here. we grow those cells compared to manual phyco isolated T cells, and both the growth and the immunophenotype match very well in both populations. Moving to the next step, T cell selection. To do that, we use CTS Dynabeads CD3 CD28 and in combination with Dynamag magnet. And the good thing for the Dynabeads is that it not only select the T cells, but also activate the T cells. So by using the Dynabeads, you achieve the selection and activation 2 in 1. And the data here shows we get very high positive recovery after T cell selection, average 93%. And those T cell subpopulation, CD4, CD8 doesn't change before processing, after processing. While we're doing this process many times, we kind of ask ourselves, can we improve this process? So in this example, we combine the PBMC isolation and the T cell selection activation step in one step, only using Rotea to process this module. In this case, we have the local pack, the incubation bag, and we add a small rocker behind the Rotea in order to do the mixing. And we have the bioreactor bag hooked on the output of the Rotea. And to start, it's the same. So we load the local pack and wash the local pack bag and transfer the cells into an intermediate bag. We inject the Dynabeads and let it incubate on the rocker for 30 minutes. And after 30 minutes, this is where it's different. Instead of hook the bag on the Dynamag, we simply load the beads and the cell complex into the chamber. And the unbound cells will be washed off while the bound cells with beads form a fluidized bed into the chamber. And then we wash this, capture the cells and beads, and eject that complex into the incubator bioreactor. And if you look at the data here, we got pretty good T cell isolation recovery, about 75%. However, the T cell purity is a little bit lower compared to those methods using the Dynamag magnets. We consider it's a kind of a quick, dirty way to isolate T cell. Of course, we are still optimizing this process. However, if you look at the T cell purity at day 3, it catch up around like 90 to 95%. Next step is wash, concentrate, and buffer exchange before electroporation. Since this step and the further downstream process step, harvest and formulation, both use Rotea, and the process quite similar, so I will use one as the example to illustrate. Here's a snapshot of cells undergo wash, concentrate, and electroporation buffer exchange. You can see the cell bed forming here, but there's not many cells showing here, but typically we would process more cells than that. And the viability from starting from the input to output typically maintains, we can see quite comparable. And we can achieve on average 86% of cell recovery in this step. After buffer exchange step, we'll go through electroporation to introduce CAR genes into the T cells. So we have two electroporation systems available. On the left here is Neon electroporation system, which is aimed for research use, R&D, or a small-scale screening. And right here we have a large volume electroporation Xenon system, which can host 1 ml single shot or 5 to 25 ml flow-through shot. And we intentionally designed both systems to be compatible, which means once you identify your perfect parameters using Neon, you can seamlessly transition these parameters into the Xenon for manufacturing purpose. Here the knock-in data we generate use both Neon and the Xenon system, targeting 3 different loci: beta-actin, Rab11A, and track locus. As you can see, the knock-in efficiency is quite comparable across the board in both Neon and Xenon system. Now we have the buffer exchange on the upstream and the electroporation Xenon on the downstream. How can we connect these two machines in order to achieve automation? So here's the example of very early prototype we are thinking of. So basically you have a conical tube sitting on a magnet with the RNP and the DNA on the tube. Once the Rotea processes the cells, it will eject the cells into the chamber. And the cells got mixed with RNP and DNA. And any residual DNA is going to be captured by the magnet. So essentially the solution is magnet-free. And then the Xenon going to draw the cell RNP and DNA complex into the Xenon electroporation chamber to do flow-through electroporation. The next step is to grow more of the CAR T cells. To do that, we have the OpTmizer serum-free medium and the immune cell serum replacement, which have been proven to provide very good T cell expansion and high viability. And more importantly, this combination involves the enrichment of T cell memory phenotype and a balanced CD4 and CD8 ratio. On the other hand, our HyPerforma Bioreactor system can further facilitate the robust T cell expansion. As you can see here, in combination with our T cell media, the T cell fold expansion is comparable to our competitor perfusion system and outperform the static system. After cell expansion, we do buffer exchange, formulation, harvest, and the final step is to cryopreserve those CAR T cells in order to be shipped back to the hospital. Our controlled-rate freezer, the CryoMed, has been extensively used in the cell therapy industry. It provides repeatable and consistent temperature performance for the final product, and it allows use of different consumables. It has a preset program for T cells, and the user can set their own protocols as well. And on the data here, we're showing after thaw, the cell can expand very well, and the viability basically before freeze and after thaw maintain above 90%. So there you have it, a complete closed workflow solution for non-viral CAR T manufacturing. And on top of that, we are actively developing the Delta-V system in order to integrate all these modules into the system so that we can achieve true automation. This is Thermo Fisher's vision for cell therapy manufacturing. First is modularity. By creating a modular system, we think it will create built-in flexibility in your process. So based on your own need, you can swap or switch different modules. And in addition, we think modularity creates efficiency in terms of manufacturing. For example, during the expansion phase, the rocker gonna expand for several days, right? So what we propose is you can simply seal off the rocker and still using the upstream modules to process other patient sample. The second is standardization. Not only we want to standardize the process step, but also by commonizing the reagent protocols and establishing in-process analytics, we want to provide the customers plug in and play, based on their individual steps. And third, the fourth point is compatibility and scalability. We want to build a system that is compatible with upstream and downstream with a variety of different machineries. And of course, the system has to accommodate for scale out in autologous manufacturing and scale up for the allogenic systems. I'll stop here, and thank you for your attention.
Thank you, Dr. Ji, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, okay, here we go. What would you define as the benefits in switching to a semi-automated manufacturing platform in this space?
That's a good question. So I think the first and the most important thing by switching to semi-automated, you limit the human errors, right? So in return, it creates the consistency of the process. And the other thing is it kind of reduce your cost, of the training, of the clean room, everything else, right? So it's definitely a benefit.
Thank you, Dr. Ji. Our next question, will the Xenon unit be GMP?
Yes, so it is designed to be GMP, but I don't have the clear timeline for it right now. I have to ask the product team. I'll get back to that.
Okay, great. Thanks again, Dr. Ji. Our next question, when will the workflow solution be available?
So right now we have the complete workflow solution, but we are working to be as a customized solution for different customers. It's not commercially available yet, but if you are interested, please contact us individually.
Okay, thanks again, Dr. Ji. Our next question, can we use non-Thermo Fisher platform on the Delta V system?
Yeah, the system I mentioned here, our main focus is only Thermo Fisher platforms. However, on the Delta V system, you can integrate other non-Thermo Fisher platforms. To do that, you have to work with the Delta V creator, which is Emerson, to incorporate those platforms.
Okay, thank you again, Dr. Ji. The next question we have. First, what would you identify as the key current bottlenecks and challenges in CAR T-cell therapy manufacturing, and how have these issues evolved over recent time?
Yeah, definitely. I mean, the current state is mostly open manual process, right? So definitely, we think create a closed system is an immediate need. So by doing that, you eliminate the contamination risk and maybe eliminate the clean room need to reduce cost as well. And the other side of it is the manual process, because this CAR-T process is really, the incoming material is really varies based on different patients. So in order to make a success manufacturing, you need to create consistency in the manufacturing process. So what we think is introducing the automation into this process will create a kind of a standard that you can do a quality control that in the end, right, you get like very, very consistent product out.
Okay, thank you again, Dr. Ji. Our next question: what are the key considerations to bear in mind when one is looking to make this switch, particularly at the earlier stages of development? What issues may arise, and how should one prepare to alleviate or address them?
Yeah, that's another good question. It's a tough question. So definitely, when we think about the design these platforms, compatibility is one issue, right? So normally people will use different companies per platform. How we gonna you gonna create a workflow that's compatible with different platforms? That's one issue. The other thing I was thinking is more of the let's say how you can reduce the need of multiple machines, right? So I think in our example here, we can use Rotea in multiple different steps. That's kind of reduce your qualification of your machine, and also with the similar machines, you reduce the training effort. So people get familiar with this machine. They have different protocols they can load at different steps. That will create a lot of efficiency.
Okay, thank you again, Dr. Ji. Our next question: how important are tools that work across multiple platforms and processes? And can you share any examples or learnings from your own experiences or workflow development success stories incorporating such tools?
Yeah, this is kind of a similar extension to what I just mentioned in the previous question. So it is very important. One example I just mentioned, the Rotea. You can use the in the PBMC isolation step, and you can use that in you wash concentrate before electroporation, and also you can use it for final formulation. So that it creates a kind of a synergy for one machine, but you can do multiple work.
Okay, thank you, Dr. Ji. Let's see, looks like we have a few more questions. How are you closing your system, and what makes it a closed system?
Yeah, that's a tough question. So what we are right now using by closing is using the as much as say stereo welding as well. So by doing that, of course, right now at the early stage, we still have to use the biosafety cabinet in order to prepare some of the reagent. But the whole process, once you load onto the machine, the only thing you do is to still weld onto different parts, or weld on or weld off.
All right. Well, thank you so much, Dr. Ji. Do you have any final comments for the audience before we go today?
Not really. I kind of covered everything, but I would just want to kind of reiterate, what we think our vision for the cell therapy field. We definitely want to create this kind of modular system, create a closed system, and standardize the whole thing. We think this will drive the cost down for CAR T manufacturing and eventually benefit the patient.
Well, thank you again, Dr. Yongchang Ji, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.